Hi, I'm Princess Senate, conference producer at Technology Networks, and I am here to introduce our next talk, Proteomics for Cancer Precision Medicine. Please join me in welcoming Dr. Janne Lehtiö as your speaker. Dr. Lehtiö is a professor at Karolinska Institute, specializing in cancer research, proteomics, and precision medicine. With a master's from Helsinki University and a PhD from KTH in Stockholm, he has extensive experience in both academia and the biotech industry. His research focuses on developing clinical applications for cancer, particularly in lung, breast, and leukemia. He has held leadership roles at SciLife Lab in Sweden and serves on various national and international advisory boards. Dr. Lehtiö is also a member of the Nobel Assembly at Karolinska Institute. Following the presentation, we will have a Q&A session and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. And with that, I will now hand over to Dr. Lehtiö to begin his presentation.
Thank you very much, and thanks for the invitation. So I'll talk about the proteomics in cancer precision medicine. So just a few introductory slides. What is precision medicine or personalized medicine? It's really adding the diagnostics and biomarker analysis to actually connect treatment for each patient. So really doing predictive analysis to see what potential treatment and drugs would be beneficial for each individual patient. So what we really need in precision medicine is the diagnostics development, but we also need the drugs to treat the patients and selection of tools, therapeutic tools as the drugs. So what does the diagnostics mean in understanding the disease and selecting treatment for patients? So I'll show you here an example of lung cancer, which I will use later also during my presentation. So lung cancer in 1990s was really considered as one disease entity, but then just looking at the morphology and histopathological features of the tumors, it was divided into disease entities such as small cell lung cancer and non-small cell lung cancer. And with a refined analysis of histopathological features, you can subdivide that into adenocarcinoma and squamous cell carcinoma and so on. And that gives a stratification of patients where you can start testing different treatments. At that time, chemotherapy and the choice of treatments for these disease entities. And then about 15 years ago, the genome medicine era brought in direct analysis of oncogenic mutations in lung cancer. And that's the first time when we can really couple treatment directly to individual patients with a treatment predictive biomarkers in a sort of precision medicine way. And that development has of course escalated a lot, and now we have a number of different mutations and number of different particular drugs connected to these oncogenic alterations. Another thing that has happened is that the toolkit of treating patients have been increased enormously by the development of new drugs and new type of treatments like cancer immunotherapies. So are we all set with this development then? Well, the problem is really illustrated in this general map of oncogenic mutations and aberrations in cancer. So basically any cancer type we analyze by genomics, we can see that it's quite difficult to find multiple drivers in one tumor. We can see few mutations and we can couple a drug potentially to these mutations. But as we know from past 100 years of oncology research, we really have multiple events in cancer that has gone wrong in order to allow a normal cell to develop into a tumor cell. That's illustrated of these well-known hallmarks of cancer, where we know that multiple events need to take place in order to turn normal cell into a malignant metastatic cancer that kills patient. So how do we actually combine these treatments to really turn the lethal cancer into a curable disease? If we look at the cancer precision medicine and genomics perspective as a breast cancer as an example, we can see the similar thing that you can find these genomic alterations, and we can take a hallmark of genomic alterations, for example, HER2 amplification in breast cancer, leading to severe breast cancer. And then we can couple HER2 inhibitory treatment to the breast cancer. And what happens is that about half of the patients respond well to the treatment and half of them don't. So this is a good news for patients that respond well to the treatment, but very bad news for the half of the patients that don't respond. And often also we get acquired resistance to the patients that respond to the treatment. So we really need to start finding rational treatment combinations using multiple hallmarks. And that's where we hit into the problems using the genomic medicine Sweden. So the poster child of genomic medicine is, for example, HER2 treatment in breast cancer, where we can see oncogenic aberrations, amplification or mutation of HER2 gene, and we can couple directed targeted treatments to HER2 to inhibit the tumor growth. However, about half of the patients don't respond to the HER2 treatment. So for the patient, that is great if the treatment is efficient, but you have also a high likelihood of non-responding. Also, if you have an early response to the treatment, often there is a resistant development. So we need to really combine treatments in order to have efficient cancer treatment. And that's where the current genomic medicine hits into difficulties. The reason for that is explained in this next slide. We have multiple somatic cancer aberrations, multiple mutations and copy number alterations that can contribute to this development of tumors, the hallmarks driving the cancer. We have also germline variants that give alter the genome regulation and the proteome regulation that can change individual mutations' impact based on the individual germline background. Then in the next level we have lots of regulation on the transcriptional and translational level, depending on chromatin structure, epigenetic regulation, and so on and so on. That leads to individual functional proteome that is related to the tumor genomic aberrations and the germline background and the regulation thereof. Then we have lots of protein turnover control and also governing the protein levels. So it's very difficult to actually predict how a particular genomic aberrations impact the functional proteome. But we treat the proteome. The fantastic new cancer drugs are actually hitting protein level. So in order to understand how do we combine these cancer drugs, we need to add proteome level information. So what we do in precision medicine nowadays is that we have added genomics very successfully into the precision medicine and cancer precision medicine. There is also transcriptomics analysis to find fusion genes and also, for example, prognostic groups in the breast cancer. Proteins have been used lots in diagnostics over the years, but the systems level proteomics is not really used in the diagnostics. Then again, tissue has been traditionally used in many types of diagnostics, especially in the cancer and the whole body imaging. So what is really missing is the actual readout of the drug targets and the molecular phenotype that the proteome networks form. So in order to really push the precision medicine into a next level, we should add the molecular phenotype level data. So if you look at the added value of refining the molecular phenotype by proteomics, it's illustrated in this slide. This is from the multi-omics study that we conducted on 140 lung cancer cases. And each dot in this picture is one protein. And by looking at the epigenetic data, transcriptomics data, and proteomics data on this cohort of 140 patients, we can see a selection of genes highlighted by the yellow circle here, where the promoter methylation really governs the transcript level all the way to the protein level. So that gene is regulated predominantly by promoter methylation. In the green circle, you can see genes that are regulated by transcriptional regulation, where the transcript level is really a good surrogate for the protein level. But in the purple circle, you can see lots of genes where the epigenetic promoter methylation state or transcript level is not very predictive of the protein level. So all those genes, the transcriptional data is very noisy. So by adding proteomics data, we can really refine the quantitative profiles and understand the molecular phenotype regulation better. So, but the real really question is there added value for proteomics in clinical decision making? What are the type of clinical decisions that we can actually try to answer by adding proteomics data? And that really needs to be shown by the data and tested in clinical trials. So I will give you some examples of our studies where proteomics can make impact. And the first study is a leukemia study where we added proteomics data, in-depth proteomics data on chronic lymphoblastic leukemia cohort, where we had genomics data, epigenomics data, also ex vivo drug screening data, and single cell data, and very well collected clinical data. And this is in collaboration with Sascha Dietrich in Germany. So what we could see from the proteomics data is that we could find very nice and defined protein subtypes of the CLL cases. So the proteomic data analysis divided the CLL in 6 subtypes. But the really interesting data really turned out to be when we looked at these subtypes in relation to the prognosis. And we can see that these subtypes are really prognostic, where the subtype 5 has actually the worst prognosis. And that subtype was very difficult to identify. The transcriptomics dataset and genomics datasets could not identify the subtype 5. So we studied further and tried to characterize the subtype 5, and that turned out to be very interesting. First, we could see that these cases of CLL cases had very high proliferation, and that was tested in the ex vivo setting where these CLL cells were growing faster than the other subtypes. Interestingly, it also had very low B-cell receptor signaling activity, and B-cell receptor signaling is known to be driving the poor prognostic CLL subtype. So this subtype seemed to be different from the previous CLL poor prognostic subtypes. It has also particular features like high spliceosomal levels and aberrant splicing activity. So these things we tested also using the phosphoproteomics and could see that the low B-cell receptor activity was also shown on the phosphoproteomics. And we could see both on the RNA sequencing data and proteomics data that there was aberrant splicing in the subtype 5. So based on this, we named this ASPCLL, Accelerated Disease Progression. High spliceosomal protein abundance is associated with aberrant splicing and low B-cell receptor signaling. So this highlights a new poor prognosis disease entity. So what we did then is that we developed a proteomics-based classifier to identify this CLL subtype using the DIA-based proteomics. To be able to do subtyping that can then be used for selection tool for clinical trials and other type of studies, and also selecting these cases, for example, for ex vivo drugs testing. So for using the proteomic subtype classifier, we then tested that in 2 new cohorts. And again, we could identify the ASPCLL subtype and we could associate that with a similar prognostic characteristics, both on time to next treatment and overall survival. Also, we could, with this larger cohort, show that these prognostic subtypes is independent of the previous prognostification tools like p53 mutation and B-cell receptor activation. So this shows the added value of proteomics in adding disease prognostification information, which is important to select maybe severe, the more harsh treatment, or trying to find cases for new type of drug treatment trials. But if we look at the other subtypes, if we look at the first 3 subtypes, we could also see a very interesting phenomenon here. If you narrow down into the first 2 subtypes, you can see that those are composed of cases where we have trisomy 12 in CLL. And trisomy 12 is a well-known aberration in chronic leukemia, where you have 3 copies of chromosome 12. But why that is enriched in leukemia, and what is the competitive advantage that the trisomy 12 gives to these leukemic cells is poorly understood. So what we also were surprised that these trisomy 12 cases were divided into 2 different subtypes, because adding one whole chromosome should change the proteome quite profoundly, and it does. But so why are they divided into 2 different subtypes? So interestingly, the trisomy 12 is divided into IGH mutated and non-mutated subtypes. And that tells about the lineage maturation stage of the CLL cells. So based on the lineage background, the trisomy 12 is divided into 2 different subtypes. So what you see in the lower part of this slide is a proteome network analysis that we use to mine the biology of these proteomics data. And you can find these protein clusters here, and you can then start annotating what are these hotspots on this protein network. And what we could see is that there is a difference between the subtype 1 and 2 in the N2 protein network, and that is actually B-cell receptor signaling, and related also to the chromosome 12 proteome. And what we could then see by detailed mining of this data, we could detect that depending on the lineage background, the maturation grade of the CLL cells, the different proteins are boosting the B-cell receptor signaling from the chromosome 12. So in both cases, the chromosome 12 added proteome actually boosts the B-cell receptor signaling. But depending on the maturation grade, it uses different proteins. So by layering the proteomics data with the genomics data, we could start understanding complex genomic aberrations and their driver event in these CLL cases. So based on this information, we re-stratify large clinical trial material on the CLL, on the clinical trial cohort in CLL. And in that clinical trial, B-cell receptor inhibitor ibrutinib was used, and by re-stratifying using our proteomics groups, we could show that the ibrutinib, the B-cell receptor inhibitor, actually wipes off the trisomy 12 boosting effect, demonstrating that our hypothesis from the proteomics data seems to be hold true. So that gives us indication that the ibrutinib can be added to the B-cell receptor or the trisomy 12 cases. So what is actually happening here, and why do we detect this on the protein level that is difficult to detect by the other levels? So if you look at this naive cartoon of mine saying on this, so you have the trisomy 12 that adds some proteins, and they are in the lineage background on unmature or more matured CLL cells. And these two forms then a synergistic impact on the protein level. So when you have this match, you can form the protein complexes that are stabilized and promoting the B cell receptor signaling pathway. And depending on the lineage background, the trisomy 12 can find different counterparts here. And that's why we see the accumulation of different protein complexes depending on the lineage background. So that's the sort of readout on the quantitative proteomics. So to summarize this part, by adding the proteomics, we could find a poor CLL prognostic subtype. We could develop proteome classifier to identify these cases prospectively. And we can also explain the trisomy 12 driver event and connect the existing treatment on this. And in altogether, we use this proteome advised CLL analysis on over 1,000 cases. So that really gives us potential to continue with this proteome advised precision medicine. So I'll show you another case where for added value of proteomics, and this is in-depth lung cancer proteomics. Also here, we used multi-omics approach to understand what is the added value of proteomics to genomics, epigenetics, transcriptomics, and also histopathological analysis of the tumors. So in this study, we teamed up with Maria Blank and Johan Staaf in Lund University and University Hospitals, and then also Åslaug Helland in Oslo University and University Hospital. Lucas Orre was driving this study in my laboratory. So we could find, again by in-depth proteomics, that this data forms 6 quite robust lung cancer subtypes. And we could confirm these subtypes with 200 additional cases. By looking at the proteomics data, we could also see that there is a groups of proteins that are very tightly correlated in quantitative terms, a little bit similarly to the CLL proteome data. This is something that we have also previously seen, and this is from our previous breast cancer proteomics dataset, where we could see this same phenomenon, that in the protein level you can see groups of proteins that are very tightly quantitatively correlated. And if you see here on the left-hand panel the protein group and the right-hand panel on the exact same data, the transcriptomics correlation of the same genes. So we started to actually look at in this breast cancer data, what are these groups of proteins? And we noticed that they are quite often protein complexes or known protein-protein interaction pairs. So we tested that in the genome-wide context, as you see on the right-hand panel. You can see random pairs and known protein-protein interaction complexes on the blue, and the quantitative correlation of the known complexes in protein level and the transcript level on blue. And you can see that the quantitative proteomics data recapitulates the protein complex data much better than the transcriptomics data. So based on this, we formed an hypothesis that we can actually use this quantitative correlation proteomics data to form protein networks to understand the proteome better. And this is actually a lung cancer network based on the 141 cases that I showed you in the first slide. Each dot here is a protein, and if they are close in this map, they are correlating. Then you can use network community detection tools to understand how is the protein communities here and to start mining the data. So in this data set, we use this for 2 different things. One is to understand the molecular phenotype of lung cancer, to mine the molecular phenotype data, and then also use the proteomics data to do unbiased cancer neoantigen analysis to understand what is the antigens that the immune system sees when it looks at the tumor. So first, if we look at the molecular phenotype of the tumors, that is the proteome network, and you can actually start mining the proteome network. What does these communities and protein groups tell us? And basically, you can draw 2 types of information from there in the solid tumors. One is that you can identify cancer-driving pathways, like mTOR pathway activation complexes, MYC pathway, and PI3 kinase pathways, and so on. But you can also see protein communities that are related to the tumor composition. You can see extracellular matrix clusters, you can see infiltration of different immune cell types and what is the state of these immune cells. You can also use this proteome network to overlay the proteomics data, quantitative proteomics data, and you can see that the subtypes feature very different hotspots in this protein network. And that directly gives you an idea that you coupling the same drugs to these different cases will not do the trick. You can also correlate the molecular phenotype data to histopathological data and do cancer driver analysis on the protein level. You can see that there is very interesting cases where you can see sky-high KRAS or EGFR or HER2 levels where we don't see the genomic aberrations, but we see a protein accumulation and can see that type of phenomenon. And then you can relate that to the histopathological features and couple the molecular phenotype to the histopathological phenotype. The immune landscape is very interesting. This is a proteomics data used to decipher the immune landscape in these subtypes. And we can see that the subtype 2 and 3 are quite immune hot, but the immune infiltration features are different between these two subtypes. We see, for example, much more B-cell infiltration and macrophage infiltration in subtype 3 compared to the subtype 2. You can also see that there is much more immune cold subtypes like subtype 4 and 5 and 6. And then you can start mining the molecular landscape of these. You can look at the antigen presenting machinery on the protein level and relate that into the subtypes. You can look at the known immune checkpoints where there is drug treatment. And this was very interesting, where you can look at the PD-1. PD-1 is an immune checkpoint that we have multiple drugs to inhibit. And interestingly, one of the immune hot subtypes, subtype 2, is sky-high on PD-1, whereas subtype 3 is low. But you can also see that there is PD-L1 high in other subtypes, and we know that the PD-L1 is not very good predictive marker for immunotherapy in lung cancer. The predictive power is not very good, so we could hypothesize that maybe if we combine the PD-L1 with the subtype 2 identification, we can have added value in predicting which patients really benefit on PD-L1 blockage. We can also recapitulate the histopathological analysis on cell infiltration by looking at the molecular phenotype data. On proteomics, we can see that we could recapitulate the B-cell infiltration patterns. So what about the cancer neoantigen profiles? So now we had genomic level data and a proteomics level data and also epigenetic screening of this. So what we use these data sets is that we use that to understand the tumor mutation burden and the microsatellite instability, which is used as estimating the potential immunotherapy response. That's the tumor mutational burden, TMB, in the first box. But in addition to that, we could analyze the cancer neoantigens by two different ways. One is detecting the testis-specific or embryonal cancer antigen by detecting the proteins that should not be expressed on the adult tissues. And that's the first panel, the CTA, that you see on the right-hand side. And you can see that the testis-specific antigen burden is varying also quite much on the subtypes. And what we also did is that we matched the peptide data back to the genome for unbiased analysis of neoantigens. And this analysis detects a very interesting group of neoantigens, pseudogenic proteins, retrovirosomal proteins, missed exon intron boundaries, where you have very antigenic long non-self peptides. And altogether we formed tumor neoantigen burden of these. And you can see quite surprisingly that the subtype 4, 5 and 6 have very high neoantigen burden, where subtype 2 has moderately high neoantigen burden. And if you remember the PD-L1 view of the tumors, it was very high on the subtype 2. So we started to mine this data and noticed that the subtype 4 and 2 differs actually on what type of neoantigens are present on those. In subtype 2, we have very high point mutation level, tumor mutation burden is high, but we have very low complex neoantigens burden. For example, these non-canonical peptides and missed exon interim boundaries, neoantigens caused by the structural variants, which is very high on the subtype 4. We could also associate that with a particular type of methylation pattern in the subtype 4. So this is interesting because we can really detect this by using the proteogenomics approach that we have developed in the group, where we actually use a data-driven way and combination of experimental way and the data-driven way to actually find with a high sensitivity these complex neoantigens. I don't have time to go into that, but we have previously published that. But what we can actually here find is completely novel classes of neoantigens. So, if we have these two subtypes, subtype 2 and 4, where we have a very high neoantigen burden in subtype 4, but very low PD-L1. So what are the features then that are contributing to this subtype? So we started to mine these on genomic aberrations and also the proteomics data. Interestingly, STK11 and KEAP1 mutations are enriched in the subtype 4. That is very interesting because these mutations are known to be related to resistance to the current PD-L1, PD-1 immune checkpoints. So that is also very well matching with the data that we shown that the PD-L1 data, PD-L1 levels are very low on this subtype. So then we started to look what is happening on the protein level and what are the outlier proteins in these subtypes. And if you look at the proteomics data, you can see that these subtypes have very different immune checkpoint expression. And interestingly, one of the major outlier proteins in subtype 4 is FGL1. And that is that at the time when we were doing this study, that was just identified as a immune inhibitory ligand of LAG3 T-cell receptor. And in this study that had just came out, they showed that in a mouse model, the high expression of FGL1 leads to low infiltration of immune cells into the tumor. And if we look at our clinical data, we can see that this is exactly the same phenotype that we could see on the subtype 4 using the proteomics data. So then we hypothesized that there is something in the subtype 4 that actually activates the FGL1 expression. And FGL1 is known to be liver-specific protein. So we turned into our proteomics data and started to mine that. And what we noticed is that there is actually several liver-specific proteins, particularly in the subtype 4 expressed. Based on that, we turned into a model system and looked at how does the STK11 mutation relate to this protein expression. And then by modulating the STK11, by reintroducing the wild type STK11 into a mutated cell line, we could see that that could then hinder the FGL1 expression, and we could also modulate the FGL1 expression by AMPK activation, which is a known effect of STK11 mutation. And then by added proteomics, we could see that the STK11 mutation actually causes activation of liver-specific transcription factor that leads to FGL1 expression and then secretion from the tumors. So to summarize this, we think that subtype 2 has lots of point mutated neoantigens, and these are not very immunogenic. compared to the long stretches of non-self peptides. So in that case, the tumor evolution and immune editing had led to the PD-L1 checkpoint, the inhibitory receptor that is enough to inhibit the T cells. But in the subtype 4, we have lots of complex neoantigens because of the chromatin state, and instead the tumors have evolved to express soluble T cell inhibitor FGL1 that diffuses from the tumors and hinders the T cell inhibitor altogether. So by layering the proteomics data, we could show actually that the STK11 mutation that is thought to activate the mTOR signaling and metabolic rewiring of the tumors also activates the immune checkpoint by inhibiting the LAG3 B cell receptor by FGL1 expression. So that way we could form a rationale for potential combination treatments. To summarize this proteomics data, we could show that the proteome subtypes again are prognostic. We could find new biological insight suggesting the drug sensitivities. And we also again developed proteome subtype classifier by high throughput proteomics and showed that we can use that in large cohorts and also late stage biopsies. So we could select patients for precision medicine using these proteomics tools. So I hope that I have shown you that the proteomics can generate added value to understand how to prognosticate and what type of treatments we can connect in cancer by using the proteomics, in addition to the genomics data. So just a few last minutes I will show to ask the question, is proteomics ready then as a biomarker tool in clinical trials? So in order to do that, we need proteomics assays that are robust and can be done on a clinical setting in a minute material. Usually we start the new treatment trials in a late stage biopsy setting. So the proteomics needs to really perform on that type of clinical setting. So based on the data that I showed you, we have developed these proteomics assays using the targeted and proteomics assays. So all of you have been in the proteomics field for a while, has understood that there is fantastic technology development. So as I've shown you now, we can use very in-depth proteomics analysis using the DDA and DIA-based proteomics, but we can also do accurate proteomics by spiking in peptides using the so-called PRM proteomics. And we can use DIA-based proteomics, which is combining features from these two methods. So we tested how does this proteomics work in a clinical setting by a small feasibility clinical trial that we called PhenoPCM. So in this trial, we set up at the Karolinska University Hospital together with a phase 1 clinical trial unit and included 25 patients across multiple tumor types. And these patients were included into the study, and we collected additional biopsies, 3 additional biopsies to test the feasibility of multiomics, adding the proteomics here. So in these biopsies were actually collected in paraffin embedded format and fresh frozen format, and we tested in the fresh frozen format by embedding them in OCT so that we could use also tissue analysis by microscopy, or we collected a fresh frozen piece. And then we tested these 3 different collections by aiming to do genomics analysis on panel sequencing and transcriptomics analysis and proteomics analysis. And this data looks really fabulous. We could do a DNA and protein level analysis of all the 25 cases using all the 3 prep methods, the FFPE and fresh frozen, And out of those, 18 got successful results on transcriptomics. So this shows that we can actually generate this type of meaningful proteomics data using clinically meaningful setting. And encouraged by that, we also teamed up with Janne Lehtiö in Oslo and did proteomics analysis on biopsies in breast cancer immunotherapy trial. And this was really a challenging task for us, because this is metastatic breast cancer where we have additional biopsy from different metastatic locations. But the proteomics data looks very, very good and robust. In most of the cases, we can identify and quantify more than 8,000 proteins. And this data looks very, very interesting in relation to the clinical response, and we are just mining that data now. So previously, we have been working on also in precision medicine tools to introduce clinical actionability annotation for genomics data using molecular tumor board portal. So this is developed in my group by David Tamburero and his team. So what we are doing here is that we have developed a molecular tumor board portal clinical tool that collects data from 7 comprehensive cancer centers across Europe in the Cancer Core Europe collaboration. And then we annotate the clinical actionability of genomic variants and present that in a patient individualized molecular tumor board report that is used in weekly meetings to sign patients for clinical trial arms. So what we are doing now is that we are working on to introduce proteomics data as part of this in SciLife Precision Medicine and initiative, together with the Karolinska University Hospital, and we are also teaming up now with Lund University Hospital and Gothenburg Sahlgrenska University Hospital and University to bring in proteomics analysis as part of the precision medicine. Also in the Science for Life laboratory, we have a large data-driven life science initiative where we try to aim to incorporate this type of data into a precision medicine report, looking at the genomics combined to the proteomics and imaging data. And this is part of our precision medicine capability, where we try to connect the data from multiple platforms at the Science for Life laboratory. We have very interesting clinical proteomics possibly at the SciLife laboratory, where we combine mass spectrometry and affinity-based proteomics in a, I would say, world unique technology platform and service platform, where we have multiple tools for affinity-based proteomics and all the tools on the mass spectrometry side that I've just presented to the data. And in particularly in the proteome medicine, we were very fortunate to get a national grant together with Karolinska Institute, Gothenburg University and Lund University to push the type of analysis on the mass spectrometry side that I just showed you in a multi-center setting. So stay tuned for our Pro-MS, Proteome Medicine Sweden initiative that we have joined forces with the Genome Medicine Sweden and Testbed Sweden to actually materialize this. And to summarize my talk, I'll show you this picture, which is actually a tube map of Paris shown by the dots here. And underneath you can see quantitative data on the peak hour traffic. So you can imagine that the tube map is like hardwired genomics data, and the peak hour traffic is the quantitative proteomics data that you can add to the hardwired genomics data. So just by layering these two information levels, you can get much more understanding what to do, where to inhibit these tumors, or where to avoid traveling in the peak hour. So I'm propagating here to get interested in proteomics, start joining forces with the proteomics people around your center, and with that, I thank you for your attention, and I'll thank my group for doing all the work. Thank you.
Thank you very much, Dr. Lehtiö. All right, so Let's kick things off with some questions already submitted from our viewers, and I suppose we'll start with a common question. How much material do you need for clinical proteomics?
That's a very good question. I would say that you can cope with a very little material nowadays. Only in a minute amount of cells or plasma can be used for clinical proteomics. So we've gone down to maybe 1,000 cells, and that gives already a very, very good data. I would say that the needle biopsy is plenty for proteomics, and so I think that we are really ready to do a clinical proteomics analysis nowadays.
Thank you. Now let's talk about speed. How fast is proteomics analysis? So in a clinical environment, I suppose time is often the essence. So understanding the time frame for analysis can be really significant. So Professor, how would you describe the typical speed of proteomic analysis in practice?
But that is also an excellent question. So that the actual proteomics analysis that we can do in a clinical setting can be very, very fast nowadays. So 20 minutes run can give you plenty of information. So the actual measurement is not the rate limiting, and that means that you can actually run lots of samples within 24 hours. Of course, it needs heavy instrumentation and some investments to the instrumentation. But the real trick is to build an organization where you can get access to the clinical samples, do the extraction and timing and move the sample to the proteomics lab, and then do the rapid data analysis and data reporting. So in genomics settings, many centers are doing clinical genomics in time scale of 2 weeks or 3 weeks, maybe the best centers within the week. So with a good organization, I think that we can definitely do that type of analysis by proteomics easily, but it's really a matter of organizing it. And at the Karolinska University Hospital, we have actually formed a particular sample preparation lab within the diagnostics division in the pathology, and we are now building a precision medicine hub where we are moving the mass spectrometry instrumentation to be able to have this rapid turnover and generate the multiomics data. And also we are working on the data reporting pipeline.
Fantastic, thank you. Before we go and end this session, I'd like to discuss another key aspect. So how would you compare proteomics data value to transcriptomics? I suppose these two fields often get compared, but I guess they each bring something unique to the table. So Professor, could you help us understand their relative values and I guess how they can work together?
Well, that's a great comparison. Transcriptomics have been used quite much in large clinical studies, and some of it is actually disseminated into the clinical routine, like detection of fusion genes and some prognostic panels, for example, in the breast cancer. I would say that this is interesting. Where I see that the proteomics really makes impact is detailed analysis on molecular phenotype, so more on a predictive side. I think that the proteomics can add very much value in prognostication, but especially to understand the treatment combinations and immunotherapy treatment prediction in cancer, understanding the molecular phenotype of rare disease or complex diseases. How does the genomic rewire the proteome, and how do we couple the treatments there? So basically, I think that we have much of the transcriptomics analysis that we have been doing in the clinical setting will actually be turning into a proteomics analysis once we get the logistics and more knowledge into the clinic, how to do proteomics, how to report the data. I think that the added value of proteomics is very high on precision medicine.
Thank you all for your fantastic questions, and a huge thank you to Professor Lehtiö for sharing his expertise with us today. It's been an absolute pleasure. Any questions that we didn't have time to get to will be sent to the speaker after the event, and as with all the other talks in the event, this talk will now be available to view whenever you want for the next 30 days. That just leaves me to say thanks again, Dr. Janne Lehtiö, and we'll be back soon with the next talk.
Thank you.